Okay, let's unpack this. Have you ever just marveled at how your body, well, works?
Right. It feels like this single coordinated thing.
Exactly, like a perfectly tuned orchestra, everything happening just right, right when it needs to. It seems so unified.
But under the hood, it's this incredibly complex web of communication. Trillions of cells constantly, talking to each other, sending signals, coordinating actions.
And it's not just nerves firing off electrical signals, is it?
Not at all. There's this whole other huge system running in parallel, a vast chemical communication network. In this chemical system, it manages, well, almost everything about how your body runs.
Everything. Like what specifically?
Oh, your basic energy levels, how fast your metabolism runs, how you grow, how you react to stress, keeping your fluids balanced, even reproduction. It's all heavily managed by these chemical signals.
Wow, so these chemicals are kind of like the conductors of that internal orchestra.
That's a great way to put it. And for this deep dive, that's exactly what we're going to explore, this really intricate world of chemical messengers.
And we've got a fantastic guide for this. We're using a really detailed excerpt from a classic physiology text.
Yeah, it's a solid source. It lays out all the different kinds of chemical communication the body uses, and it really zooms in on the hormone systems, which are, the powerhouses.
So what's our mission today? What are we trying to achieve with this deep dive?
We're aiming to go deep, like really deep, not just skimming the surface. We want to truly understand, to master the fundamental principles of how these chemical messengers work, especially the hormones.
Mastering it like the pros do.
Exactly. Getting down to the core concepts, how these molecules are actually built, their structure, how they get around the body, how they deliver their signals with such incredible precision.
And even how scientists figured out ways to measure these tiny, tiny amounts of powerful stuff.
Right. Think of it as building the absolute foundational knowledge. The bedrock. Everything else in endocrinology kind of rests on understanding these fundamentals.
Okay, sounds like a plan. Where do we start? Broad overview first.
Yeah, let's start broad. Look at the whole spectrum of chemical signals the body uses. Then, just like our source does, we'll zero in on the hormones, the endocrine and neuroendocrine systems, and really dig into their complexity and, frankly, their importance. Ready?
Shit, dive in.
Okay, so the body coordinates all its countless activities using multiple types of chemical messenger systems. It's definitely not just one size fits all.
Different systems for different jobs, I guess.
Precisely. Speed, reach, target, it all varies. Our source identifies 6 key types of these chemical communicators.
6. Okay, that already tells you how crucial this is. What are they?
First up, probably the most familiar for many people, neurotransmitters.
Right, like dopamine, serotonin, that sort of thing.
Exactly. These are chemicals released by the very end of a neuron, the axon terminal. But here's the key.
Yeah.
They're released into a tiny, tiny gap called the synaptic junction, right next to the target cell.
So super localized.
Extremely localized. Their action is fast, very targeted, and they control the functions of the nerve cell, muscle cell, or gland cell right on the other side of that synapse. Think speed and precision over incredibly short distances.
Like a direct private message between two cells right next to each other.
Perfect analogy. Okay, second type. Endocrine hormones. This is where our main focus will be, based on the source.
The classic hormones like insulin or thyroid hormone.
Exactly. These are produced by specialized glands, thyroid, pancreas, pituitary, adrenals, and so on, or sometimes by specialized cells scattered in other tissues. They release their hormones directly into the circulating blood.
Ah, into the bloodstream, so they travel.
They travel all over the body. The blood carries them far and wide, allowing them to influence target cells that might be located a long, long way from where the hormone was actually made.
So unlike the private chat of neurotransmitters, this is more like a public broadcast announcement sent out via the body's highway system.
That's a great way to think of it. A broadcast message available to any cell that has the right receiver to pick it up.
We'll definitely come back to those receivers. Okay, neurotransmitters are local, endocrine hormones are broadcast. Is there something in between?
There is, and it's fascinating. Type 3, neuroendocrine hormones.
Neuroendocrine.
Sounds like a mix.
It's exactly that, a hybrid. These hormones are actually secreted by neurons, just like neurotransmitters. But instead of releasing them into a synapse,
They release them into the blood.
bingo, they release them directly into the circulating blood. So they originate from the nervous system, but they travel and act like endocrine hormones, influencing target cells at distant locations.
Wow, that's a direct link between the nervous system and the hormone system.
A crucial bridge. Our source gives key examples. ADH, or vasopressin, and oxytocin. Both are made by neurons in the hypothalamus, but released into the bloodstream from the posterior pituitary gland.
And another really important group are the hypophysiotropic hormones, also made by hypothalamic neurons. But these are released into a special blood vessel system, the portal system, in the median eminence, and they travel just a short distance to control the anterior pituitary gland. It shows neurons directly controlling hormone release via the blood.
So the brain can basically tell glands far away what to do using these hormone signals carried by the blood. That interplay seems really important.
It's fundamental to homeostasis.
Okay.
Moving on. What about signals that act locally, but aren't necessarily neurotransmitters at a synapse and don't travel far in the blood?
Like neighborhood communication.
Exactly. That brings us to type 4, paracrines. These are secreted by cells into the fluid just outside them, the extracellular fluid.
Okay.
And they act on neighboring target cells, but crucially, these target cells are usually of a different type than the cell that released the signal. They just diffuse through the local interstitial space.
So not a broadcast, not a private synapse message, but like yelling over the fence to your neighbor.
Huh. Yeah, that works. Talking to the nearby, but different neighbors. Then, type 5, autocrines.
Auto, like self.
Precisely. These are also secreted into the extracellular fluid, but they act back on the very same cells that produce them.
The cell talks to itself. Why would it do that?
It's a form of self-regulation or feedback. The cell releases a signal, and that signal binds to receptors on its own surface, telling it maybe to slow down production or ramp up or change its activity based on what it just released.
Okay, so paracrine is talking to the neighbors. Autocrine is giving yourself instructions or feedback. Makes sense. What's the last one? Number 6.
Number 6 is cytokines. These are peptides, so chains of amino acids, secreted by cells, especially immune cells, but others too, into the extracellular fluid.
Peptides, like the protein hormones we talked about.
Similar structure, yeah. But the key thing about cytokines is their versatility. They can act as autocrines, talking back to the cell that made them. They can act as paracrines, signaling nearby cells. And sometimes they can even enter the bloodstream and act as true endocrine hormones traveling to distant targets.
Whoa, they can do it all. The jack-of-all-trades chemical messenger?
Pretty much. They're really flexible. Classic examples are interleukins and lymphokines in the immune system. Helper T cells release them to signal other immune cells nearby or sometimes further away.
Okay.
The source also mentions a specific term, adipokines. These are cytokine hormones, like leptin, that are produced specifically by adipocytes, or fat cells. Leptin travels through the blood to the brain to regulate appetite, so it acts like an endocrine hormone in that case.
So cytokines are defined by being peptides, but their function, auto, para, or endocrine, depends on the context where they're released and where the receptors are.
You got it. And as we said, our source, and therefore our deep dive today, is going to focus primarily on those endocrine and neuroendocrine systems, the ones involving blood transport for wider communication.
The broadcast and the neuron-initiated broadcast systems?
Right. But it's super important to remember, as you pointed out earlier, these systems don't operate in isolation. They're constantly interacting.
The source had examples, right? Like the adrenal glands.
Yeah, perfect example. The adrenal medullae, the inner part of the adrenal glands, secrete epinephrine and norepinephrine. But what triggers that release? Often it's direct stimulation from nerve signals from the sympathetic nervous system.
Nerve signal causes hormone release. Direct neuroendocrine link.
Exactly. Same with the pituitary gland. Its hormone release is heavily controlled by signals from the hypothalamus, many of which are those neuroendocrine hormones we discussed. It's a constant back-and-forth dialogue between the nervous and endocrine systems to keep everything in balance, to maintain homeostasis.
Okay, so focusing on these blood-borne endocrine hormones, they travel everywhere, but you mentioned they don't affect every cell. It comes down to the receiver.
Absolutely. The specificity of hormone action relies entirely on receptors. Hormones exert their effects by binding to specific protein receptors located either on the surface or inside their target cells. If a cell doesn't have the receptor for a particular hormone, that hormone will just float on by, having no effect on that cell, no matter how much of it is around.
The receptor is the gatekeeper, the lock that only the right hormone key can open.
That's the perfect way to think about it. And our source makes an important distinction here about reach. Some hormones, like growth hormone or thyroid hormone, tyrosine, have receptors on many, many different cell types throughout the body.
So they have widespread effects.
Right. Growth hormone promotes growth pretty much everywhere. Thyroxine increases metabolic rate in almost all cells. But most hormones are more targeted. They act primarily on specific target tissues,
Why only specific tissues?
because those tissues have a much higher abundance of the specific receptors for that hormone compared to other tissues. Think of ACTH, adrenocorticotropic hormone. From the pituitary, it mainly stimulates the adrenal cortex because that's where its receptors are highly concentrated. Or ovarian hormones, which primarily affect female reproductive organs and tissues involved in secondary sexual characteristics because those tissues are packed with estrogen and progesterone receptors.
So the message goes everywhere via the blood, but only cells with enough of the right receptors really hear the message and respond strongly. That specificity is critical.
Fundamental. And this brings us right back to the so what? Why should you care about mastering these hormone systems?
Because they run the show.
Pretty much. The source is very clear. Hormones regulate almost all major body functions. Metabolism, how you use energy, growth and development, water and electrolyte balance, reproduction, digestion, even aspects of behavior. It's all under significant hormonal control.
The examples in the text really drive this home.
They do. Imagine life without growth hormone. You simply wouldn't grow to a normal size. Without thyroid hormone, your metabolism would crawl, you'd feel incredibly sluggish, maybe gain weight easily. Without insulin from your pancreas, your cells can't use sugar properly. That's diabetes.
And the sex hormones.
Without the right sex hormones, sexual development wouldn't happen normally, and reproductive function would be impossible. These aren't just minor adjustments. They're essential drivers of what we consider normal life and health. Understanding them is understanding your body's core operating system.
Okay, that definitely underscores the importance. They're fundamental. So we know that they signal and roughly where they act based on receptors. Let's get into the nitty-gritty. What are these messengers actually made of? What's their chemical nature? And how does that influence how they're produced?
Excellent question. It turns out hormones fall into pretty distinct chemical categories, and as you guessed, their chemical makeup is absolutely key. It dictates how they're synthesized, how they're stored, or if they're stored, and how they get released. Our source identifies three major chemical classes.
Three main types of chemical structures for hormones.
Yep. And interestingly, the source explicitly states there are no known hormones made of polysaccharides, complex carbohydrates, or nucleic acids like DNA or RNA.
Okay, so no sugar-based or gene-based hormones. What are the three classes then?
The first, and by far the largest class, is proteins and polypeptides.
So chains of amino acids.
Exactly. This category includes a huge number of hormones. Think of almost all the hormones from the anterior posterior pituitary gland, the pancreas, like insulin and glucagon, the parathyroid gland, parathyroid hormone or PTH, and many, many others listed in tables and physiology texts.
What's the difference between a protein and a polypeptide here? Just size.
Basically, yeah. The source uses a common rule of thumb. Polypeptides generally have fewer than 100 amino acids, while proteins have 100 or more. Hormones in this class span that whole range. You have tiny ones like TRH, thyrotropin-releasing hormone, with only 3 amino acids, all the way up to big ones like growth hormone or prolactin, with close to 200 amino acids.
Okay, so variable length chains of amino acids. How does a cell actually build these? Is it the standard way proteins are made?
It is. It follows the central dogma, pretty much. Synthesis starts on the ribosomes attached to the rough endoplasmic reticulum, ER, but they usually don't start as the final active hormone.
They start as something else.
Yeah, typically they're first synthesized as larger inactive precursor molecules called pre-pro hormones. Think of it like a first draft with extra bits.
Pre-pro hormone, okay.
Then, inside the ER, an enzyme usually clips off a signal peptide part, turning the pre-pro hormone into a slightly smaller, but often still inactive prohormone. Second draft still needs editing.
Okay, pre-pro hormone to prohormone in the ER, then what?
From the ER, the prohormone gets shuttled over to the Golgi apparatus. The Golgi is like the processing and packaging center. Here, the prohormones are sorted and packaged into tiny membrane-bound sacs called secretory vesicles.
So they get put into little delivery packages. Is that where the final editing happens?
Often, yes. It's typically inside these secretory vesicles, as they bud off the Golgi and mature, that enzymes perform the final cut. They cleave the prohormone into the final biologically active hormone molecule. Sometimes this cleavage also produces other fragments which might be inactive or have their own functions. But the key is the active hormone is now ready inside the vesicle.
So the finished product is ready to go, sealed in its package. Where do these packages wait?
These vesicles, filled with the active hormone, are then stored in the cytoplasm of the endocrine cell. Often they gather near the cell membrane, waiting for the signal to be released.
And how do they get released? They can't just diffuse out, right, the proteins.
Correct. They're generally too large and water-soluble to just slip through the membrane. Their release happens via a process called exocytosis, when the cell gets the right stimulus.
Like a nerve signal or another hormone?
Exactly. When stimulated, the vesicles move to the cell membrane, fuse with it, and then basically spill their contents, the active hormone, and any fragments outside the cell, usually into the nearby capillaries to enter the bloodstream.
And what's the internal trigger for that fusion, that exocytosis?
It often involves an increase in the concentration of calcium ions inside the cell cytoplasm. This calcium influx can be triggered by various signals, like a change in the cell membrane's electrical voltage, depolarization, or, very commonly, by the activation of signaling pathways linked to receptors on the cell surface. These pathways might involve other messengers like cyclic AMP, cAMBP, which can activate protein kinases, enzymes that then phosphorylate proteins involved in vesicle fusion.
So a signal comes in, triggers calcium or CANAPE, which activates machinery to make the vesicles dump their cargo.
You've got it. And once released, because these are peptides and proteins, they are generally water-soluble.
Which means they dissolve easily in the blood plasma.
Yep, makes the transport straightforward. They just dissolve in the plasma and get carried around the body to their target tissues, where they can easily discharge out of the capillaries into the surrounding fluid.
Okay, that's the big group. Proteins and polypeptides. Made like proteins, stored in vesicles, released by exocytosis, travel dissolved in blood. What's the second major chemical class?
The second class is steroid hormones. These have a very different chemical nature.
Steroids, like cortisol, testosterone, estrogen.
Exactly those. They're secreted by the adrenal cortex, cortisol, aldosterone, the ovaries, estrogen, progesterone, the testes, testosterone, and also the placenta during pregnancy, estrogen, progesterone.
And chemically, they're not proteins at all.
Not at all. They're derived from cholesterol. Their basic structure is that characteristic 4-ring structure, 3 6-carbon rings and 1 5-carbon ring fused together. Because they're built from cholesterol, they are highly lipid-soluble, meaning they dissolve well in fats and lipids, but not well in water.
Fat soluble. Okay, that sounds like it would make a big difference in how they're handled compared to water-soluble peptides.
A huge difference. Let's start with synthesis and storage. Because they are lipid-soluble, steroid-producing cells generally store very little of the actual finished hormone.
They don't store the final product in vesicles like peptide hormones.
Nope. Instead, they often store large amounts of the precursor, cholesterol, often in the form of cholesterol esters in lipid droplets. When the cell receives a signal to produce the steroid hormone,
It quickly converts the stored cholesterol.
right, a complex series of enzymatic reactions happening in the mitochondria and ER converts cholesterol step by step into the specific final steroid hormone, like cortisol or testosterone. The rate of synthesis is the main control point.
And once it's made, how does it get out? No exocytosis if it's not in a vesicle.
Exactly. Because they are so lipid-soluble, they don't need vesicles or exocytosis. As soon as the finished steroid hormone is synthesized, it simply diffuses right across the lipid bilayer of the cell membrane, out into the interstitial fluid, then into the nearby blood capillaries.
Just slips right through.
Yeah.
Yeah.
Release is basically immediate upon synthesis. The amount released is determined by how fast the cell makes it, which is controlled by the stimulating signals the cell receives.
So, peptide hormones make, package, store, release on demand via exocytosis. Steroid hormones store precursor, make on demand, release immediately via diffusion. Very different logistics.
Fundamentally different. And the cholesterol precursor, it can either be synthesized de novo within the cell itself, or the cell can take it up from cholesterol circulating in the blood, usually carried by LDL particles.
Okay, that covers steroids. What's the third chemical class?
The third class is derivatives of the amino acid tyrosine. These are also called amine hormones.
Derived from just one amino acid, tyrosine, not a chain like peptides.
Correct. They're relatively small molecules formed by enzymatic modifications of tyrosine. This class includes 2 very important groups of hormones, the thyroid hormones and the catecholamines secreted by the adrenal medulla.
Thyroid hormones, T4 and T3, and the fight-or-flight hormones, epinephrine or epinephrine.
Exactly those 2 groups. They are both made from tyrosine by enzymes located in the cytoplasmic compartments of the glandular cells. However, how they are stored and released is quite different between the 2.
Okay, let's take thyroid hormones first. How are they handled?
Thyroid hormones, thyroxine, T4, and triiodothyronine T3 have a unique storage mechanism. They are synthesized within the thyroid follicular cells, but then they are incorporated into a very large glycoprotein molecule called thyroglobulin.
They get attached to another big protein.
Yes, and this thyroglobulin, loaded with T4 and T3 molecules, is then secreted and stored outside the cells in the central lumen of the thyroid follicles, forming what's called colloid. So the thyroid gland stores a large supply of its hormones extracellularly within its own structure.
Stored outside the cell, that's different. How are they released then?
When the thyroid gland is stimulated by TSH from the pituitary, the follicular cells take up some of the thyroglobulin from the colloid by endocytosis. Inside the cell, lysosomes fuse with these vesicles, and enzymes digest the thyroglobulin, cleaving off the T4 and T3 molecules. These free T4 and T3 hormones, being lipid-soluble, can then diffuse out of the cell and into the bloodstream.
Wow, complex process. Synthesized, attached to a scaffold, stored outside, brought back in, cut loose, then released. And transport in blood. Are they water or lipid soluble?
They behave like lipid-soluble hormones in transport. Once in the blood, they bind extensively to plasma proteins, especially thyroxine-binding globulin, TBG. This protein binding is crucial, as we'll see.
Okay. Now, what about the other tyrosine derivatives, the adrenal medulla hormones, epinephrine, adrenaline, and norepinephrine, or adrenaline?
These catecholamines are also synthesized from tyrosine via a series of enzymatic steps within the adrenal medullary cells. The adrenal medulla normally secretes about 4 times more epinephrine than norepinephrine.
And how are they stored and released, like thyroid hormones or like peptides?
Unlike peptides, once synthesized, epinephrine and norepinephrine are taken up into preformed vesicles within the cytoplasm of the adrenal medullary cells and stored there.
Stored inside, in vesicles, so releases by
Exocytosis, just like peptide hormones. When the adrenal medulla receives a nerve signal from the sympathetic nervous system, these vesicles fuse with the cell membrane and release their contents, epinephrine and norepinephrine, into the bloodstream.
Okay, so tyrosine derivatives share a precursor, but thyroid hormones are unique, stored extracellularly on thyroglobulin, lipid-soluble transport, while adrenal catecholamines are handled more like peptides, stored in vesicles, released by exocytosis, water-soluble transport.
Exactly. Understanding these 3 classes, proteins, peptides, steroids, and tyrosine derivatives, and their distinct synthesis, storage, and release strategies is really fundamental. It sets the stage for everything that follows. How they travel, how long they last, and even how they interact with cells.
Which is the perfect place to go next. We know how they're made and launched. Let's talk about their journey and their lifespan. How do they get where they need to go in the vast circulatory system? And how does the body make sure the signal doesn't last forever? Control the duration.
Right. The timing is critical.
Right.
And hormones vary massively in how quickly they act and how long their effects last. It's all tailored to the job they need to do.
Like, give me the extremes.
Okay. Norepinephrine and epinephrine, the catecholamines, can be secreted and exert their full effects within seconds to minutes. Think of the rapid heart rate increase during a scare. That's them.
Super fast.
Then you have hormones like thyroxine or growth hormone. Their effects might take days to become noticeable and can take months to reach their full potential, regulating long-term things like overall metabolic rate or body growth.
Seconds versus months. That's an incredible range. It really shows their specialized tools. Now, you mentioned earlier they circulate in tiny amounts. How tiny are we talking?
It's truly mind-bogglingly small. Hormone concentrations in the blood are typically measured in picograms, that's 10-12 grams, to, at most, a few micrograms, 10 to 6 grams per milliliter of blood.
Picograms, trillionths of a gram per milliliter. How can amounts that small possibly have such powerful effects on the whole body?
That comes down to the amazing sensitivity and amplification systems built into the target cells. The receptors and the signaling pathways they trigger are designed to respond powerfully to even these minuscule concentrations. A little goes a very long way.
Okay, so tiny amounts, but powerful effects. How do they actually travel in the blood plasma, which is mostly water? Does it depend on their chemical class?
Absolutely. It comes down to water solubility versus lipid solubility. The water-soluble hormones, that's the peptides, proteins, and the catecholamines, epinephrine or norepinephrine, have no problem. They just dissolve directly in the plasma.
Like sugar dissolving in water?
Exactly. They get carried along passively by the blood flow from where they're secreted to the target tissues. When they reach the capillaries near their target cells, they can easily diffuse out through capillary pores into the interstitial fluid and reach to the cell receptors.
Simple enough for them, but what about the lipid-soluble ones, the steroids and thyroid hormones? They don't like water.
Right. They face a challenge dissolving in watery plasma. So they circulate mainly bound to plasma proteins. These are carrier proteins like albumin or specific globulins like thyroxine-binding globulin, TBG for thyroid hormones, or corticosteroid-binding globulin, CBG, for cortisol.
So they hitch a ride on these proteins.
They do. Our source emphasizes that usually less than 10% of these lipid-soluble hormones are circulating freely dissolved in the plasma at any given time. For thyroxine, it's even more extreme. Over 99% is typically bound to protein.
Wow, almost all of it is tied up. Does the bound hormone do anything?
Not directly. Hormones bound to these plasma proteins are generally considered biologically inactive. They can't easily diffuse out of the capillaries and reach the receptors on target cells because the protein complex is too large. Only the tiny fraction that is free, unbound in the plasma, is biologically active and can exert effects.
So why bind them up then? What's the point?
It serves a couple of crucial functions. First, it acts as a reservoir. The large amount of bound hormone circulating provides a ready supply. As the small amount of free hormone gets used up by tissues or cleared from the body, some of the bound hormone dissociates from the carrier protein to replenish the free pool. This helps buffer changes and maintain a relatively stable concentration of the active free hormone.
Like a reserve tank ensuring a steady supply to the engine. Makes sense. Does it affect how long they stick around?
Absolutely. This protein binding dramatically slows down the clearance of these hormones from the plasma.
How so?
Because the bound hormone is protected, it's less available for breakdown by enzymes in the blood or tissues, and it's less likely to be filtered out by the kidneys, since the large protein complex doesn't easily pass through the kidney filters.
Ah, so being bound keeps them safe from removal.
To a large extent, yes. Contrast that with the water-soluble hormones circulating freely. They're much more exposed to degrading enzymes and readily filtered by the kidneys. Our source gives a striking example. Angiotensin II, a water-soluble peptide, has a half-life in the blood of less than a minute.
Gone in under 60 seconds. That's incredibly fast clearance.
It is perfect for something involved in rapid blood pressure adjustments. But the protein-bound hormones, they last much longer. Adrenal steroids might have half-lives of 20 to 100 minutes, and the heavily protein-bound thyroid hormones can persist for 1 to 6 days.
Days. That fits perfectly with their role in regulating slower, long-term processes. So this binding is a key factor in determining the duration of the hormone signal.
A major factor. Now, how does the body actually measure this rate of removal? There's a specific term for it.
Right, you mentioned it earlier, the clearance rate.
The metabolic clearance rate, or MCR. It quantifies how quickly the body removes a hormone from the plasma. It's usually expressed in milliliters of plasma cleared of the hormone per minute.
Okay, ml of plasma per minute? How do they figure that out?
Conceptually, the formula is MCR equals the rate at which the hormone disappears from the plasma divided by the hormone's concentration in the plasma.
Okay, but how do you measure the disappearance rate in a living system?
The source describes a common experimental method. You take a purified sample of the hormone you're studying and tag it with a radioactive isotope. Then, you infuse this tagged hormone into the bloodstream at a constant known rate. You keep infusing until the concentration of the radioactive hormone in the blood plasma reaches a steady state, meaning the rate you're infusing it equals the rate the body is removing it, the disappearance rate.
Ah, inflow equals outflow, steady level.
Exactly. At that point, you measure the concentration of the radioactive hormone in the plasma. Now you know the disappearance rate, which equals your infusion rate, and the plasma concentration. Plug those into the MCR formula, and you can calculate the clearance rate.
Very elegant. So once we know the MCR, what are the actual ways the body gets rid of these hormones? Where do they go?
Several routes. The source lists the main ones. 1. Metabolic destruction by tissues. Enzymes in various tissues, both target and non-target, like liver, kidney, blood, can break down the hormone molecule, inactivating it. 2. Binding with tissues. Hormones can be taken up by cells and effectively removed from circulation, even if not immediately destroyed. 3. Excretion by the liver into the bile. The liver is a major player, especially for steroid hormones. It often modifies them, EZF conjugation, to make them more water-soluble, then excretes them into the bile, which eventually leaves the body in feces. The source notes liver disease can mess this up, causing hormones to build up. 4. Excretion by the kidneys into the urine. The kidneys filter blood. Small water-soluble hormones, or hormone metabolites made by the liver, that aren't bound to proteins can be filtered out and excreted in the urine.
So breakdown in tissues, uptake by tissues, disposal via liver bile, disposal via kidney urine, multiple pathways.
Yep. And there's one more important site of clearance mentioned, degradation at the target cell itself.
The cell receiving the signal also helps clean it up.
Sometimes, yes. After a hormone binds to its receptor on the cell surface, the entire hormone receptor complex can be internalized by the cell through endocytosis. Once inside, enzymes within the cell can break down the hormone molecule. Often, the receptor part is then recycled back to the cell membrane, ready to bind another hormone molecule.
Efficient. Use the signal, dispose of the messenger, recycle the receiver. This whole system of transport and clearance seems incredibly well managed to ensure signals start and stop appropriately.
It absolutely is. The rapid clearance of some hormones allows for quick on-off switching or pulsatile signaling, while the slow clearance of others allows for sustained, steady effects. It's all part of the fine-tuning.
Okay, this is fascinating. We've covered what they are, how they're built, how they travel, how long they last, and how they're removed. Now, the real magic, the mechanism of action. Once that tiny amount of hormone finally reaches its target cell, how does it actually tell the cell what to do? How does that binding event get translated into a specific cellular response?
This is where it gets really intricate and powerful. And it always starts with that first step. The hormone has to bind to its specific receptor. No binding, no action.
The key in the locks.
Exactly. And these receptors are typically large protein molecules. A single target cell might have thousands, even up to 100,000 receptors for a particular hormone. Crucially, each receptor type is usually highly specific for just one hormone. This ensures that even with dozens of different hormones circulating, a cell only responds to the ones it's supposed to.
Specificity again. And we talked about receptor location being key. Where are they found again?
Right. Their location fundamentally determines how the signal gets relayed inside the cell. Our source outlines 3 main locations based on the hormone type. 1. On the cell surface membrane. This is where you find receptors for the water-soluble hormones, the proteins, peptides, and catecholamines. They can't cross the lipid membrane, so they need an external receptor to pass the message along. 2. In the cell cytoplasm. This is the primary location for receptors for most steroid hormones. Steroids are lipid-soluble, so they easily diffuse through the membrane to find their receptors waiting inside. 3. In the cell nucleus. Receptors for the thyroid hormones are mainly found here. The source suggests they're often directly associated with the chromosomes. Thyroid hormones are also lipid-soluble, crossing the cell and nuclear membranes to reach these nuclear receptors.
Okay, so water-soluble hormones signal from the outside in. Lipid-soluble hormones go inside to cytoplasmic or nuclear receptors. Is the number of these receptors fixed? Or can the cell adjust how sensitive it is?
Definitely not fixed. This is a super important concept. Regulation of receptor number and sensitivity. Target cells can dynamically change how strongly they respond to a hormone by altering the number of active receptors they have, or by changing the sensitivity of the signaling pathways downstream. It's not static at all.
The cell can turn the volume up or down on the hormone signal.
Exactly. One major way is downregulation. If a cell is exposed to a really high concentration of a hormone for a prolonged period, it often responds by decreasing the number of active receptors available for that hormone.
Why would it do that?
It's usually a protective mechanism to prevent the cell from being overstimulated or overwhelmed by excessive signaling. It makes the target tissue less responsive to the hormone.
Like becoming desensitized. Or how does the cell physically reduce the receptors?
Several ways, according to the source. It might chemically inactivate the receptor proteins themselves. It might inactivate some of the intracellular signaling molecules that link the receptor to the response. It could temporarily pull the receptors inside the cell, hiding them from the surface, sequestration. It might destroy the receptors more quickly using lysosomes after they're internalized, or it could simply slow down the production of new receptor proteins. Lots of options.
And can it go the other way? Can a cell become more thrombocytic?
Yes, that's upregulation. In some situations, the stimulating hormone itself, or other signals, can cause the target cell to increase the number of active receptors or to enhance the activity of the intracellular signaling components. This makes the target tissue progressively more sensitive to the hormone's effects.
Priming the pump, so to speak.
Sort of, yes. So this ability to modulate receptor number and function is a crucial layer of control, allowing cells to adapt their responsiveness to changing hormonal environments.
Okay, so the hormone binds its specific receptor, and the cell can even adjust how many receptors it has. What happens next? After that initial binding, this is the intracellular signaling part.
This is where the signal truly gets transmitted and amplified within the cell. The formation of that hormone receptor complex kicks off a cascade of biochemical reactions inside.
And this is where a tiny signal gets magnified.
Absolutely. A single hormone binding event at the surface can lead to the activation of many enzyme molecules inside, which in turn activate even more molecules, leading to a final response that's exponentially larger than the initial trigger.
How does that work for the receptors on the cell membrane? How do they get the signal across?
Good question. The source briefly mentions ion channel link receptors, where binding opens or closes an ion channel, changing ion flow across the membrane. This is common for neurotransmitters, less so for direct hormone action, although some hormones might indirectly affect ion channels via other receptor types.
Okay, so ion channels aren't the main way for hormones at the membrane. What are the big players?
A very common mechanism involves G-protein-linked hormone receptors. These receptors are amazing proteins that snake back and forth across the cell membrane 7 times.
7 transmembrane domains. I remember seeing diagrams of those.
Exactly. And on the inside surface of the membrane, these receptors are coupled to a family of helper proteins called heterotrimeric G proteins. Heterotrimeric just means they have 3 different subunits: alpha, beta, and gamma.
So the receptor talks to this G protein trio.
It does. In the G protein's inactive state, the alpha subunit is bound to a molecule called GDP, guanosine diphosphate, and all 3 subunits are huddled together. Now, when a hormone binds to the outside of the G protein-linked receptor, the receptor changes shape.
The binding causes a conformational change.
Right. This activated receptor can now interact with the inactive G protein complex nearby on the inner membrane surface. This interaction triggers a key event in the G protein. The GDP molecule pops off the alpha subunit, and a GTP molecule, guanosine triphosphate, takes its place.
Swapping GDP for GTP. That's the on switch for the G protein.
That's the critical activation step. Once GCP is bound, the alpha subunit carrying the GTP detaches from the beta and gamma subunits. This newly freed active alpha GTP subunit can then move along the inner membrane surface and interact with other membrane-bound proteins, which are the effectors.
Effectors. Like enzymes.
Often enzymes, yes. Common effectors are enzymes like adenylate cyclase or phospholipase C. Sometimes the effector can be an ion channel. When the active alpha subunit bumps into and binds its specific effector protein, it changes the activity of that effector, turning it on or sometimes off. This change in effector activity is what initiates the next step in the intracellular signaling cascade.
Okay, hormone binds receptor. Receptor activates G protein by GTP swap. Alpha subunit activates an effector enzyme or channel. How does the signal stop? Does the alpha subunit stay active forever?
No, it has a built-in off switch. The alpha subunit itself has enzymatic activity. It's a GTPase. It can slowly hydrolyze the bound GTP back to GDP.
It cuts off its own power source.
Basically, yes. Once GTP is cleaved back to GDP, the alpha subunit becomes inactive again. It then lets go of the effector protein and reassociates with the beta and gamma subunits, reforming the inactive G protein trio, ready for the next signal. It's a self-regulating cycle.
Neat. A built-in timer.
And there's more complexity. The source mentions there are different types of G protein alpha subunits. Some are stimulatory, Gs. When activated, they increase the activity of their effector, like activating adenyl cyclase. Others are inhibitory, G. They decrease the activity of their effector, like inhibiting adenyl cyclase.
So it's the same hormone binding to different receptor types coupled to Gs versus G could have opposite effects inside the cell.
Exactly. It provides enormous flexibility and allows for very fine-tuned regulation of cellular processes. Many hormones use these G protein-coupled receptors.
Okay, that's a major mechanism for membrane receptors. What's the other main type?
The other big category is enzyme-linked hormone receptors. As the name suggests, these receptors either are enzymes themselves or they're very tightly associated with enzymes on the inside of the cell that they activate upon hormone binding.
So the receptor itself does the enzymatic work or calls over an enzyme partner?
Correct. Unlike the 7-pass G protein receptors, these typically only pass through the membrane once. When the hormone binds to the part sticking outside the cell, it activates the enzymatic domain or the associated enzyme partner on the part inside the cell.
What kind of enzymes are we talking about?
A very important class is the receptor tyrosine kinases, RTKs. The receptor itself has tyrosine kinase activity in its intracellular domain. Binding of the hormone, often growth factors like insulin, IGF-1, or VEGF, causes the receptors to dimerize, pair up, and phosphorylate each other on tyrosine residues, which then activates their kinase domains to phosphorylate other target proteins inside the cell.
Okay, the receptor itself is the kinase. What about the associated enzyme type?
Our source gives the leptin receptor as a great example of this type. Leptin is that hormone from fat cells we mentioned. Its receptor doesn't have intrinsic kinase activity. But when leptin binds, the receptor subunits come together and activate a separate associated tyrosine kinase from the JAK family, specifically JAK2, that's bound to the receptor's intracellular domain.
So the receptor acts like a scaffold to bring JK2 kinases together and turn them on.
Precisely. Once activated, JK2 phosphorylates tyrosine residues on the receptor itself and also on other signaling proteins called STATs, signal transducers and activators of transcription.
Stats sound like they do something with genes.
They do. Phosphorylated STATs move into the nucleus and act as transcription factors, binding to DNA and activating the transcription of specific genes. This leads to the synthesis of new proteins, which mediate some of the slower, longer-term effects of leptin.
Slower effects via gene transcription. Does JK2 do anything faster?
Yes, activated JK2 also triggers other faster signaling pathways like the MAPK pathway and the PI3K pathway, which involve cascades of protein phosphorylation leading to more immediate cellular responses. So one receptor can launch both slow, gene-based, and fast phosphorylation-based signals.
That's really complex signaling from one hormone binding. The source also mentioned adenyl cyclase in this enzyme-linked section.
Yes, sometimes the receptor itself can be adenyl cyclase, the enzyme that makes CAMP. While often activated via GS proteins, some receptors might have this activity intrinsically or be tightly linked. This enzyme is crucial because CAMP is a major second messenger, which we need to talk about next. Similarly, some receptors, like the one for atrial natriuretic peptide, are linked to guanylate cyclase, making another second messenger, cGMP.
Okay, so membrane receptors use G proteins or enzyme links to initiate the signal cascade inside. What about those lipid-soluble hormones, steroids, and thyroid hormones that go inside the cell to find their receptors? How do they work? No G proteins or membrane enzymes there.
Correct. Their mechanism is fundamentally different. They rely on intracellular hormone receptors and gene activation.
So they interact with DNA?
Directly or indirectly, yes. Remember, steroids and thyroid hormones and related molecules like vitamin D and retinoids are lipid-soluble. They easily diffuse across the cell membrane and enter the cytoplasm or even the nucleus. Inside, they seek out and bind to their specific intracellular receptor protein.
Which could be waiting in the cytoplasm or already in the nucleus.
Right. If the receptor is in the cytoplasm, common for many steroid hormones, hormone binding often triggers a conformational change that unmasks a nuclear localization signal. The hormone receptor complex then gets transported into the nucleus. If the receptor is already in the nucleus, typical for thyroid hormones, the hormone just needs to get there and bind it.
So the activated hormone receptor complex ends up in the nucleus one way or another. Then what?
Once in the nucleus, this activated complex functions as a transcription factor. It binds to specific short sequences of DNA located near the genes that the hormone regulates. These DNA binding sites are called hormone response elements, HREs.
They bind right onto the DNA code.
Yes, and this binding event directly influences the rate at which that specific gene is transcribed into messenger RNA, mRNA. The complex might activate transcription, turn the gene on or up, or sometimes repress transcription, turn the gene down or off.
So they control which genes get read.
Exactly. The mRNA produced then travels out of the nucleus to the cytoplasm, where ribosomes translate the mRNA code into new proteins.
So the ultimate effect is making new proteins.
Precisely. These newly synthesized proteins, which could be enzymes, structural proteins, transport proteins, etc., are what carry out the actual cellular response directed by the hormone.
Can you give an example?
The source uses aldosterone, a steroid hormone that acts on kidney tubules. Aldosterone diffuses into the tubule cell, binds its receptor, mineralocorticoid receptor in the cytoplasm. The complex goes into the nucleus, binds to HREs on the DNA, and switches on the genes for proteins like sodium channels and the sodium-potassium pump. These new proteins get inserted into the cell membrane and increase the kidney's ability to reabsorb sodium and excrete potassium.
And because it involves making new proteins from scratch, transcription, translation, this takes time, right?
Absolutely. That's a key feature of steroid and thyroid hormone action. There's typically a lag period of maybe 45 minutes to several hours or even days before the full physiological effect is seen, because you need time to synthesize enough of the new proteins. Much slower than the near instantaneous effects you can get with hormones acting via second messengers at the membrane.
What about thyroid hormones? Similar idea.
Very similar mechanism. But the receptors are typically already in the nucleus, often bound to DNA even before the hormone arrives. Thyroid hormone, key 3 is more potent, binds to these nuclear receptors, activating them to stimulate the transcription of a large number of genes. The source mentions potentially over 100 different proteins.
Wow, 100+ proteins.
Yeah, many of which are enzymes involved in increasing metabolic activity throughout the body, and their effects are also long-lasting. Once bound, thyroid hormones can influence gene expression for days or weeks.
And you mentioned something really interesting earlier, that even if different tissues have the same intracellular receptor, they might respond differently. How does that work?
That comes down to tissue-specific gene regulatory proteins. While the hormone receptor complex might bind to the same HRE sequence near a gene in different cell types, whether that gene actually gets turned on or off strongly depends on the presence and activity of other nearby co-activator or co-repressor proteins. These helper proteins are often expressed differently in different tissues. So the cellular context, the unique set of other regulatory proteins present, determines the ultimate transcriptional outcome.
So the receptor provides the who, which hormone, but the cell's internal environment provides the which specific genes get regulated. That's incredible specificity.
It really is. It allows a single hormone to orchestrate very different but appropriate responses in different parts of the body.
Okay, we've covered how the signal gets initiated, both at the membrane and inside the nucleus. For those membrane-acting hormones, you kept mentioning second messengers. Let's really break those down. How does the signal actually propagate inside the cell after that initial receptor activation?
Right, this is crucial for understanding how most peptide and catecholamine hormones work. The hormone itself, the first messenger, binds to the receptor on the outside, but generally doesn't enter the cell. Its job is done once it activates the receptor. That activation then triggers the production or release of small intracellular signaling molecules called second messengers. These are the guys that actually spread the signal within the cell and trigger the downstream effects.
So the hormone knocks on the door and the second messenger answers and runs around inside delivering the message.
Excellent analogy.
Yeah.
And they also amplify the signal significantly. Our source highlights three really important second messenger systems. The first, and maybe the most widely used, is the cyclic adenosine monophosphate, CMP system.
CMP. We mentioned adenyl cyclase makes it, often activated by GS proteins.
Exactly. So hormone binds receptor activates the JS protein,
Okay.
Midigen BF protein, activates the enzyme adenyl cyclase on the intermembrane surface. Adenyl cyclase converts ATP, the cell's energy currency, into CAMP. Now you have CAMP inside the cell.
What does CMP do?
Its main job is to activate another enzyme called CAMP-dependent protein kinase, which is often called protein kinase A or PKA.
Kinase. That means it phosphorylates things, adds phosphate groups to proteins.
Precisely. Activated PKA goes around phosphorylating specific target proteins within the cell. This phosphorylation acts like a molecular switch, turning the activity of these target proteins, which are often other enzymes or regulatory proteins, either on or off. This change in protein activity then leads to the cell's ultimate response to the original hormone.
And the amplification happens here too.
Oh yeah, big time. Remember that cascade? One hormone molecule, multiple G proteins, multiple adenyl cyclases, lots of CMP molecules. Each CAMP activates PKA. Each PKA can phosphorylate many target proteins. Each phosphorylated protein might be an enzyme that catalyzes many reactions. It's amplification at multiple stages, leading to a massive response from a tiny initial signal.
That explains the potency. And what if the hormone receptor links to an inhibitory G protein, G?
Then the opposite happens. Activated G inhibits adenyl cyclase. Less adenyl cyclase activity means less CAMP is produced. Lower CMP levels mean less PKA activation, leading to an inhibitory effect on the cell compared to baseline or GS stimulation.
So the same system can stimulate or inhibit just by swapping the G protein linkage.
Exactly. And critically, the final outcome of changing CAMP levels depends entirely on the target cell.
How so?
Because different cells have different sets of proteins that PKA can phosphorylate. The source gives great examples. In thyroid cells, CAM-PKA activation leads to thyroid hormone synthesis. In adrenal cortex cells, it leads to steroid hormone secretion. In kidney tubule cells, it increases water permeability via aquaporin channels. The second messenger, CMPG, is the same, but the cellular machinery it acts upon dictates the specific response. Many hormones use this pathway: ACTH, glucagon, TSH, LH, FSH, PTH, vasopressin via V2 receptor, catecholamines via beta receptors. The list is long.
Fascinating how one internal signal drives such diverse actions.
Yeah.
Okay, what's the second messenger system?
The second one involves calcium ions, Ca2+, and the protein calmodulin. In this system, the initial hormone signal leads to an increase in the concentration of free calcium ions within the cell's cytoplasm.
Where does the extra calcium come from?
Two main sources. It can enter the cell from the outside fluid through calcium channels in the plasma membrane. These channels might be opened by a change in membrane voltage, or they might be linked somehow to the hormone receptor activation. The other major source is release from internal stores within the cell, primarily the endoplasmic reticulum or sarcoplasmic reticulum in muscle and mitochondria.
So calcium floods into the cytoplasm either from outside or from internal tanks, and that calcium surge is the signal.
Yes, normally cytoplasmic calcium levels are kept incredibly low, like 100 nanomolar, 10 to 7 m. A signal might cause them to rise tenfold or even 100-fold to 10.6 or 10.5 m. This rise in calcium is the second messenger signal, but calcium usually doesn't act alone. It exerts many of its effects by binding to an intermediary protein called calmodulin.
Calmodulin, what does it do?
Calmodulin is a small protein found in basically all cells.
Yeah.
It has 4 binding sites for calcium ions. When calcium levels rise, calcium ions bind to calmodulin. When 3 or 4 of these sites are occupied, calmodulin changes its shape and becomes activated.
Okay, calcium binds, calmodulin activates.
This activated calcium-calmodulin complex can then interact with and regulate the activity of many other proteins, especially enzymes. A key target group is the calmodulin-dependent protein kinases. Just like pKa, these kinases phosphorylate specific target proteins, triggering the cellular response.
So calcium rise binds calmodulin complex, activates kinases. Kinases phosphorylate targets icellular response. Similar logic to the CAMP pathway, but using calcium as the trigger.
Very similar logic. Phosphorylation cascades are a common theme. The source gives smooth muscle contraction as an example. The calcium calmodulin complex activates myosin light chain kinase, which phosphorylates myosin, leading directly to muscle contraction. It also notes the similarity between calmodulin and troponin C in skeletal muscle, which also binds calcium to trigger contraction. Cool connection. Okay, so CATMP and calcium calmodulin are 2 big ones. What's the third system the source highlights?
The third involves the breakdown products of membrane phospholipids, specifically a phospholipid called phosphatidylenosyl biphosphate, PIP2.
Breaking down fats in the membrane creates signals.
Exactly. Certain hormone receptors, when activated, turn on an enzyme called phospholipase C, PLC, which is located at the inner surface of the membrane, often activated via a G protein, specifically GQ. PLC's job is to find PIP2 molecules in the membrane and cleave them into 2 separate second messenger molecules.
2 signals from one cut. What are they?
They are inositol triphosphate, IP3, and diacylglycerol, DAG.
IP3 and DO, what do they do differently?
They have distinct roles. IP3 is small and water-soluble, so it diffuses away from the membrane into the cytoplasm. Its main target is specific IP3-gated calcium channel receptors located on the membrane of the endoplasmic reticulum. When IP3 binds to these receptors,
It releases calcium from the internal stores. Bingo. IP3 binding opens those channels, causing a rapid release of stored C2+ into the cytoplasm. This surge in cytoplasmic calcium then acts as a second messenger, often working via calmodulin, as we just discussed, to trigger responses like muscle contraction or cell secretion.
So this pathway directly links to the calcium signaling pathway. What about the other product, beta?
DAG is lipid-soluble, so it stays put, embedded in the cell membrane. Its main job is to activate another important kinase called protein kinase C, PKC. Activated PKC then phosphorylates a whole different set of target proteins compared to pKa or calmodulin-dependent kinases, leading to a wide range of cellular responses specific to PKC action.
Okay, so PLC activation valley cleaves PIP2 into IP3 and DAG. IP3 goes to the ER area, releases calcium calmodulin effects. DAG stays at the membrane, activates PKC phosphorylates other targets. 2 branches from one starting point.
Precisely. And the source adds another layer. The lipid part of DAG is often arachidonic acid. This can be further processed by other enzymes to produce prostaglandins and related local hormones, eicosanoids, which have their own widespread effects. So this pathway can branch out even further.
Wow, that's really intricate signaling. Which hormones use this PLC-IP3D system?
The source lists several, including angiotensin II, acting on certain receptors, catecholamines, acting via alpha receptors. Note this is different from the beta receptors that use CAMP. GnRH, oxytocin, and vasopressin, acting via its V1 receptor, again, different from the V2 receptor that uses CAMP. It really highlights how one hormone can trigger different internal signals depending on the specific receptor type it binds to in a particular cell.
That truly demonstrates the flexibility and complexity. Hormone hits receptor, triggers internal relays using CAMP or calcium calmodulin or IP3-DIG, activating kinases, Phosphorylating proteins may be changing gene expression, leading to the final action. Understanding these pathways is clearly essential to grasping how hormones work at a molecular level.
It absolutely is. Mastering these intracellular signaling concepts is fundamental to understanding hormone action and, by extension, physiology and pathophysiology.
The detail here is amazing, really helps achieve that deep understanding we're aiming for. But stepping back again, all this knowledge is based on being able to actually measure these things. How on earth did scientists figure out how to detect hormones when, as you said, they're present in like picogram amounts? That seems almost impossible with standard chemistry.
It was a massive challenge for decades. Early endocrinology was mostly about observing effects, remove a gland, see what happens, inject an extract, see what happens. They couldn't accurately quantify the actual hormone levels in blood or tissues because the concentrations were just far too low for traditional chemical methods.
So what changed? What was the breakthrough?
The absolute game changer was the development of radioimmunoassay, or RIA, by Rosalind Yalow and Solomon Berson back in 1959. Yalow actually received the Nobel Prize for this work. RIA provided the incredible sensitivity needed to measure these minuscule amounts.
Radioimmunoassay. Okay, sounds like radioactivity and antibodies are involved.
Exactly. The core principle relies on two things. One, having an antibody that binds very specifically and tightly to the hormone you want to measure. And two, using a fixed amount of that same hormone that has been tagged with a radioactive isotope as a competitor.
A radioactive competitor? How does that work?
Okay, here's the setup. First, you need that highly specific antibody against your hormone. Then you take your sample, say blood plasma, which contains an unknown amount of the natural hormone. You mix this sample with two other things: a small, limited quantity of the antibody and a known fixed amount of the purified hormone standard that has been radioactively labeled.
So you have natural hormone, unknown amount, radioactive hormone, known amount, and not enough antibody to bind all of them.
That's the crucial part. The antibody concentration is limiting. Because there aren't enough binding sites on the antibodies for all the hormone molecules, natural plus radioactive, they have to compete for those sites.
Ah, competition.
Yes. The natural hormone from your sample and the radioactive standard hormone are essentially fighting for the available spots on the antibody. The amount of each that successfully binds will be proportional to how much of each was present in the mix.
So if my sample had a lot of natural hormone, it would outcompete the radioactive one for the antibody spots.
Exactly. A high concentration of natural hormone means more natural hormone binds, leaving less room for the radioactive hormone to bind. Conversely, if your sample had very little natural hormone, the radioactive hormone would win most of the binding spots.
Okay, I see the inverse relationship. Less radioactive bound means more natural is present. How do you measure that?
After you let the mixture reach binding equilibrium, you need to separate the antibody molecules, which now have some mixture of natural and radioactive hormone bound to them, from the rest of the solution containing the unbound hormones. There are various ways to precipitate or capture the antibody complex. Then you take that separated antibody hormone complex and measure the amount of radioactivity associated with it using a gamma counter or a similar device.
So you measure the radioactivity that got bound. How do you get from that radioactivity count to an actual concentration of the natural hormone?
You need a standard curve. You run the exact same procedure, but instead of using your unknown sample, you use a series of standard solutions containing known, progressively increasing concentrations of the non-radioactive natural hormone. For each known concentration standard, you measure how much radioactive hormone gets bound to the antibody. You then plot these results. Known natural hormone concentration on the x-axis versus the amount of radioactivity bound on the y-axis.
And because of the competition, as the known natural hormone concentration goes up, the radioactivity bound should go down.
Exactly. You get an inverse relationship curve. Once you have this standard curve, you take the radioactivity measurement from your unknown sample, find that value on the y-axis of your standard curve, and read across to the x-axis to determine the corresponding concentration of the natural hormone that must have been in your sample.
That is incredibly clever. Using a radioactive tracer in competition to indirectly measure something present in vanishingly small amounts. And the sensitivity is high enough for picograms.
Easily. RAA can measure down into the picogram, 1012G, or even femtogram, 1015G, per milliliter range. Accuracy is typically pretty good, maybe 10–15% error. It completely revolutionized endocrinology research and clinical diagnostics. Suddenly, you can measure hormone levels accurately.
Is RIA still the main method used today? Or have things moved on?
RIA is still used, but another technique called enzyme-linked immunosorbent assay, or ELISA, has become extremely widespread, especially in clinical labs. It offers similar sensitivity and specificity, but has some practical advantages.
ELISA. I've heard of that one too, often used for detecting antibodies or antigens. How does it work for measuring hormones? Does it also use competition?
Some ELISA formats can use competition, but the most common type for hormone measurement, often called a sandwich ELISA, usually doesn't rely on competition. And crucially, it uses enzymes instead of radioactivity for detection.
Enzymes instead of radioactivity? Okay, how does the sandwich ELISA work?
It's often done in 96-well plastic plates, which is great for automation. Here's the basic idea, as described in our source. First, the wells of the plate are coated with a specific antibody, let's call it the capture antibody, AB1, that binds to the hormone you want to measure. This antibody sticks to the plastic surface. 2. Then you add your samples or standards containing the hormone to the wells. The hormone, if present, gets captured by binding to AB1. You wash away anything unbound. 3. Next, you add a second antibody, the detection antibody, AB2. This antibody also binds specifically to the hormone, but to a different spot on the hormone molecule in AB1. So the hormone gets sandwiched between AB1, stuck to the plate, and AB2. Wash away unbound AB2. 4. Now comes the enzyme. You add a third antibody, AB3, that is designed to recognize and bind to the detection antibody, AB2. And this AB3 is chemically linked to an enzyme, like horseradish peroxidase or alkaline phosphatase. So AB3 enzyme binds to the AB2 part of the sandwich, wash away unbound AB3 enzyme. Finally, you add a chemical substrate, S, that the enzyme attached to AB3 can act upon. The enzyme converts the substrate into a detectable product, P. This product is often colored or fluorescent or chemiluminescent. 6. You then measure the amount of product formed using an optical reader, like a spectrophotometer or a fluorometer.
Whoa, that's a multi-layer sandwich. AB1 captures hormone, AB2 binds hormone, AB3 enzyme binds AB2. The enzyme is the final reporter.
Exactly. And the key to ELISA sensitivity is enzyme amplification. One single molecule of enzyme captured in that sandwich can churn through thousands or millions of substrate molecules, generating a huge amount of detectable product over time. This massively amplifies the signal from even a tiny amount of captured hormone.
So, unlike RIA's competition, ELISA often relies on this enzyme amplification, and the amount of product is related to the hormone amount.
Yes, in a typical sandwich ELISA, you use an excess of the antibodies,
Yeah.
AB1, AB2, AB3. The goal is to capture all the hormone present in the sample. Therefore, the amount of enzyme that gets bound in the well, and thus the amount of colored fluorescent product generated, is directly proportional to the initial concentration of the hormone in the sample. More hormone, more sandwich, more enzyme, more signal.
Direct relationship, not inverse like RIA. That seems simpler to interpret. What are the big advantages of ELISA?
The source highlights a few key ones. No radioactivity is a big plus: safer, easier handling, less regulatory hassle, no radioactive waste disposal. The 96-well plate format makes it very easy to automate, so labs can run lots of samples efficiently and cost-effectively. And ELISAs are generally very sensitive and accurate, making them a robust and reliable method for quantifying hormones and many other proteins.
So ELISA built on the antibody specificity idea from RIA, but swapped radioactivity for enzyme amplification and streamlined it for modern labs. These measurement techniques seem absolutely fundamental to everything we've discussed. They're what allowed the field to move from just observation to precise quantification.
Couldn't have said it better myself. Without RIA and then ELISA, our understanding of endocrinology wouldn't be nearly as deep or sophisticated as it is today. They were and are cornerstone technologies.
This has been an absolutely fantastic deep dive. We've really dug into the fundamental ways your body communicates using chemicals, zeroing in on hormones exactly as our source material guided us. We've covered the whole landscape of chemical messengers.
Right, distinguishing the local messengers like neurotransmitters, paracrines, and autocrines from the long-distance travelers, the endocrine and neuroendocrine hormones that rely on the bloodstream.
And we spent a lot of time on the hormones themselves, their chemical families. The peptides proteins, the cholesterol-derived steroids, and the tyrosine-based amines. We saw how their chemistry dictates how they're made, stored, and released.
Which in turn affects how they travel, whether dissolved freely or bound to carrier proteins, acting as reservoirs and influencing how long they stick around, governed by their metabolic clearance rate.
Then we got into the real core, the mechanisms of action. The critical role of specific receptors on the membrane for water-soluble hormones, inside the cell for lipid-soluble ones, and how the cell can even regulate the number and sensitivity of these receptors.
And we dissected those intricate Intracellular signaling pathways for lipid-soluble hormones going right to the nucleus to control gene transcription and protein synthesis, leading to slower but sustained effects.
And for the membrane-acting hormones, we unpacked those amazing second messenger systems, the cascades triggered by G proteins or enzyme-linked receptors using internal relays like CAMP or calcium calmodulin or the IP3-DAG duo derived from membrane lipids.
Highlighting how these systems amplify the initial signal incredibly, allowing tiny hormone concentrations to exert massive cellular effects with the specific outcome tailored by the unique machinery within each target cell.
And finally, we appreciated the ingenuity behind the measurement techniques, RIA and ELISA, that allow scientists to actually detect and quantify these powerful molecules despite their minuscule concentrations. This really feels like building that foundational mastery-level understanding we set out to achieve.
It does, and when you step back and look at it all, what's truly striking is just the sheer elegance and precision of it all. It's breathtaking, really.
Elegance? In what way?
Just the way it's all put together. The precise enzymatic cleavage of prohormones, the dynamic up and down regulation of receptors to fine-tune sensitivity, the complex interlocking second messenger pathways that allow for amplification, diversification, and integration of signals. The way different hormone structures are perfectly matched to different transport needs and durations of action. It's like a masterwork of biological engineering, honed over millennia.
When you put it like that,
Yeah.
yeah, it's not just a collection of parts. It's a deeply integrated, responsive system.
Exactly. And connecting this back to the bigger picture, to you, understanding these fundamental principles isn't just academic. It's the absolute foundation for understanding health and disease.
How so?
Think about it. Diabetes is fundamentally a problem with insulin signaling. Thyroid disorders involve too much or too little thyroid hormone action. Growth problems often relate to growth hormone pathways. Issues with stress response involve cortisol signaling. Reproductive health relies entirely on the correct functioning of sex hormone pathways. Almost every major physiological process, and consequently many diseases, involves these chemical signaling systems going awry at some level.
So mastering these basics is key to understanding what goes wrong in so many conditions. It really highlights the delicate balance required for normal function.
It absolutely does. Which leads to maybe a final provocative thought to leave you with.
Okay, let's hear it.
Given this incredible complexity, this intricate web of receptors, G proteins, kinases, second messengers, feedback loops, all working in concert, how vulnerable might this exquisitely tuned system be?
Vulnerable to what?
To disruption. Think about external factors, environmental chemicals, components of our diet, chronic psychological stress, pharmaceuticals designed to target one part of a pathway. How easily might these subtly interfere with specific steps in these signaling cascades? And if one small part of this interconnected network gets knocked out of sync, what are the potential cascading consequences throughout the body?
That's a sobering thought. That the very complexity that makes it work so well might also make it susceptible to subtle interference with potentially wide-ranging effects.
Something to definitely ponder as you go about your day while your body silently, constantly manages its own internal chemical symphony.